Hey Fungi friends, I'm back to read you chapter 7 from my book, Passport to Kingdom Fungi. So this is kind of a sciencey chapter. This is one where I'm flexing my muscles a little bit, using my background as a PhD biochemist to try to make sense of both medicinal mushrooms as well as toxic mushrooms, and then kind of dive into the science behind them. And a reminder that I wrote this book very much as kind of a pushback to some of the pop science mycology that happens where people state their opinions as facts but don't always back them up with scientific citations or actual evidence to kind of support what they're saying. And so I did a lot of research to write this book. And in particular, this chapter was one where I dove really deep into scientific literature and was trying to look at like what was a real thing versus what was kind of hype. And hopefully I did a good job of that. And I even did some original sort of research and like ran a whole experiment with the help of a friend for this chapter to add some data to the sort of raging debate between fruiting bodies versus mycelium when it comes to like medicinal mushrooms. So I'm going to go ahead and read this, but I want to remind you guys that I'm doing this for everyone who wants to sort of hear and read the audio version of the book. I don't have a sponsor. I'm not getting paid. I am promoting my book and potentially asking you to buy the book if you enjoy what I'm doing. That is a great way to support me. But this is all about accessibility, and that was the whole point of doing this. Is just that I wanted my book to be read and heard and experienced by as many people as possible. And so far, it's been pretty difficult to get the book out there. It was released last year in 2025, and it didn't get a whole lot of press, and it didn't get onto like the greatest, best science books of 2025. It wasn't part of any reader lists. It unfortunately, I don't know if people pay for that stuff or people just get lucky. It's been a lot slower going trying to promote this book than I thought it would be. And it's, I have some positive momentum, and I'm really grateful for the incredible opportunities I've been given to present on my book and talk about it in a public sphere. But it's been tough, and it's been tough on me personally too, because this is sort of my whole life. I vested everything I had in my career and my future in this book, and it's been difficult to get traction and build that momentum. So again, if you enjoy this and you like listening to me talk about the book, please pick up a copy of Passport to Kingdom Fungi. Recommend it to your friends, family, etc. If you like this podcast / video series, please feel free to share it with your friends and family as well, and also comment or give it a rating because that helps other people find this. So with no more ado, I'm going to get reading. This is Chapter 7 from Passport to Kingdom Fungi by me, Dr. Gordon Walker. And this is the Fungal Biochemistry, the Science of Medicinal and Toxic Fungi. So I'm going to try to do my best to read everything here, and occasionally when I'm speaking quickly, I skip things or don't pronounce stuff right. So bear with me if that happens, but I'm doing my best to read to you guys in a calm and hopefully informative manner. So fungi are incredible chemists with the ability to produce a dizzying array of complex molecules as a natural part of their growth, many of which serve a dual purpose in modulating their biology and ecology. Fungi are also capable of making a vast array of proteins and enzymes that catalyze the transformation of their food, keeping competitors at bay and modify their local environment. So fungi are just constantly producing stuff that is kind of chemically altering the world around them, and that's part of what I'm saying here. Since fungi live in their food, they don't have an easy way to dispose of waste. So many of their metabolic waste products are concentrated in the externalized reproductive structures we know as mushrooms. So a lot of that secondary metabolites that mushrooms produce as part of detoxifying stuff or just as part of their growth cycle ends up in the mushroom. That's kind of how mushrooms are the toilet for the mycelium, if you get what I mean. Fungi are nature's pharmacy, containing a bevy of beneficial bioactive compounds that modern medicine has used to develop drugs that have saved countless lives. However, in addition to life-saving drugs, fungi can also produce a variety of toxins that can make you severely sick or even end your life if you're not careful. And I will talk about those at the end of the chapter. What are primary and secondary metabolites in fungi? Primary metabolites are the core molecules involved in fundamental cellular processes like growth, development, and reproduction. Secondary metabolites are usually more complex molecules that require a set of specialized enzymes and a multi-step sequential chemical pathway to be produced. So basically, primary metabolites are just the stuff that like fungi are eating and breathing all the time. And then when they get to the more complex chemistry, those are what are called secondary metabolites. Well, secondary metabolites are not directly involved in growth or reproduction. They often benefit the organism producing them. That's why they make them. Secondary metabolites tend to be expensive. Oh, here, I talked about that. Okay. Secondary metabolites can be energetically costly to produce, but they serve important roles, helping to mediate ecological interactions, providing a selective advantage against competition, as well as facilitating communication with plants, animals, and other fungi. Endophytic fungi, in conjunction with bacteria, produce a wide range of secondary metabolites that can guide plant growth through regulating hormone production and producing an array of bitter metabolites that deter insects from feeding. In fact, a lot of plants that have like herbs or anything that's bitter or has a strong aroma is often being somewhat manipulated or driven, puppeted by endophytes inside of the plant. And those are usually bacteria and fungi. And sometimes it's the interaction of both the bacteria and the fungi that puppet the plant. And those things can be like upregulated or downregulated depending on all sorts of factors. But it's really cool. A lot of the stuff we attribute to plants may actually be due to the fungi and the bacteria that are living inside of plants, which is part of what makes endophytes so incredibly interesting. And they are by far doing some of the coolest chemistry out there. So infectious pathogenic strains of Aspergillus produce gliotoxins, which allow them to propagate by suppressing the immune system of their host. And so that's an example of something that Aspergillus is doing to, infect stuff. It's a secondary metabolite, gliotoxin, that it's producing. A majority of the biologically active and medicinally relevant compounds found in fungi are secondary metabolites. Okay. And I have a little like diagram here of various types of fungal metabolites. I'll read a couple to you just as examples. Peptides, one that is made by cordyceps is called cordyheptapeptide, and it's got anti-cancer and tumor activity, sort of a big complex molecule. There's one called ergotamine made by claviceps, and that deters animals from feeding. That is a precursor to lysogenic acid, LSD. Speaking of which, indole alkaloids, so psilocybin, made by psilocybe mushrooms and a couple other genre of saprotrophic mushrooms. And so those are, in theory, made to be anti-herbivory, deter animals from feeding, can act as an antioxidant as well. There's a cool one called xylidin. It's a kind of quinone, and that's made by chlorosiboria, the green elf cups, and it's like a green pigment that's in wood, and it turns the wood green. But they also use it as like an optoelectronic material. So I was like digging through all the different metabolites in fungi and finding all sorts of cool examples of stuff, and that's a few of what I put into this little diagram here. So I'll keep reading. What are medicinal mushrooms? Many mushrooms are considered medicinal because they contain compounds associated with positive health outcomes. Specifically, these mushrooms tend to contain a range of primary and secondary metabolites with bioactive properties like polysaccharides, polyphenols, proteins, terpenes, sterols, organic acids, and more. Many of these compounds have been tested under laboratory conditions and demonstrated anti-inflammatory, antimicrobial, antiviral, and anti-cancer properties, as well as the ability to lower cholesterol and modulate our immune system, among other beneficial effects. So there's stuff in fungi that can do all sorts of things. And that is clear that fungi have a ton of bioactive molecules that can have really amazing like medical effects. The real question there is, do those actually get into us in a biologically available form and at a high enough concentration to have those effects like in the human body? But I'll get to that. While mushrooms and other fungi contain a broad range of bioactive compounds, here we go. The concentration of these compounds in mushrooms, extracts, or supplements may not be high enough to be medically relevant. Moreover, these compounds are often not bioavailable, meaning our bodies cannot absorb them in their natural state. The potential of medicinal mushrooms is promising, but it's important to keep in mind that the evidence for many of these effects has primarily been demonstrated in the lab with model organisms like mice rather than in clinical studies with human subjects. So that's exactly what I was saying. You have to always sort of be somewhat skeptical every time there's a new paper that says, look, this mushroom contains this molecule and it does this thing to cancer. Because sure, in a really high concentration, like a test tube or on a petri dish, you can get a particular activity, but the question is, does that transfer to the human body, or is it medically relevant when, if you're just eating a mushroom, or do you have to like take that compound and turn it into a drug or a medicine and then deliver it to specific tissue to get the same activity? Okay, what should I know if I want to consume medicinal mushrooms? Some common edible mushrooms like lion's mane or hericium, maitake, grifola, shiitake, lentinula, are highly regarded for their medicinal properties. However, many medicinal mushrooms like reishi, ganoderma, turkey tail, trametes are inedible due to their hard, woody texture and require special preparation for consumption. Cooking, drying, and powdering whole mushrooms is, in my opinion, the best way to access the full spectrum of polysaccharides and bioactive compounds. However, decoctions and tinctures made by alcohol water double extractions have also been demonstrated to contain relevant bioactive compounds. Generally, fungi fruiting bodies contain a higher concentration of bioactive compounds than the mycelia. Thus, products made from mushrooms are more potent by weight than products made from mycelia grown on grain. That being said, the chemical compositions of products made with fruiting bodies versus mycelia can be different, each providing specific benefits. For example, lion's mane mushrooms contain hericenones, while the mycelia contain ericenes. Both compounds have been shown to stimulate nerve growth in mice, but erinacines are more bioavailable since they cross the blood-brain barrier. And there was a whole sort of study done out of Taiwan that was showing that, because they were comparing the two compounds for their activity together. And I think in like a straight assay, they had pretty similar activity. But when you actually wanted to get them absorbed into the organism, the erinacines are the ones that actually cross the blood-brain barrier, and so they were able to get into the brain and ideally cause the activity in the brain. And so as much as I am usually on the side of saying that fruiting bodies are probably better because they have more bang for your buck in terms of the bioactive compounds, in the case of lion's mane, if you're trying to get that nerve growth activity, it might actually be beneficial to take the mycelium powder instead. And what I've heard from people sort of anecdotally is that for people who have neurodegenerative diseases, whether it's Parkinson's or ALS or dementia or something, lion's mane is not going to cure it. There's no cure for that kind of thing. But maybe in a very careful sense, I've heard a lot of anecdotal stuff that says maybe lion's mane can slow down the progression of the disease. And so what I've seen is people who have suffered traumatic like nerve injuries or have these neurodegenerative diseases and start taking lion's mane, you have to take a lot. It's not like taking one pill a day. You have to take something like 5 to 10 grams of lion's mane mycelium every day. And the best way to put that down is often in some sort of like smoothie or shake or something like that. And that is not going to, again, cure anything, but it might slow things down a little bit. And I have talked to people who've also managed to I'm not going to say cure, but get back to normal after like big injuries. And they said that lion's mane has helped them. So I'm skeptical of all this stuff, but I've also talked to a lot of people who've been very much helped by it. Conversely, there's also a whole community of people out there, especially on Reddit, who say that lion's mane has hurt them for all sorts of different reasons. And I don't necessarily believe that either. I'm kind of skeptical of both the people who say it's like an amazing cure and then the people who say that it has hurt them. And I'm like, it's just a mushroom and the mycelium somewhere in between. So I'm always on the side of like, let's gather more data before we make really definitive statements. But I'm just telling you guys a little bit more about what I've heard, and I didn't have space to write about that stuff in the book. Okay, this is a good one. Are mushroom supplements safe and effective? Despite their potential health benefits, medicinal mushroom supplements are unregulated natural products. Currently, no mushroom-based products have official approval from the Food and Drug Administration for any prescribed medicinal purpose. In 2020, the FDA issued warning letters to several companies selling mushroom supplements advertised as having curative properties requiring them to remove the associated health claims. And you'll see this. A lot of mushroom supplements now have very sort of like weak language around what it can do for you because it's quite illegal to actually tell people that it'll like cure your ADHD. That doesn't stop a bunch of brands from using that kind of marketing, but they're not allowed to. There are FDA-approved pharmaceutical drugs derived from fungi, but these have been purified, tested, and the dosage is well defined. However, there can be benefits to consuming mushroom products. The entourage effects from consuming the full spectrum of bioactive compounds in mushroom preparations can act together to potentially exert synergistic positive effects. It's called the entourage effect, and it's similar in cannabis in that it's not just like the THC that gets you high, it's also the terpenes and all the other stuff. And so if you're taking mushrooms as medicine, you can have a single compound that has been isolated, and if you are delivering that to a certain tissue at a certain dosage, you may be getting a defined effect. However, you might be missing out on some potential synergistic effects of having like a whole mushroom extract or having a whole mushroom period and having multiple things kind of stack on top of each other. And there's not a lot of science to necessarily support that, but it makes a lot of sense biologically, which is why I mentioned it in here. Keep in mind that the inherent complexity of compounds in mushrooms makes it nearly impossible to predict all of the positive or negative effects they can have on an individual. And so that's a really important thing to remind yourself of too, is that mostly we would view the idea of taking medicinal mushroom products as being positive for our health, net positive. But there may be certain situations where it could be negative, and either you take too much of something or you take something that inhibits like another drug or something you're taking, which is why it's always a good idea to talk to a doctor about, oh, hey, I'm going to add this mushroom supplement. And they might say, oh, you know what, we've heard about negative interactions between this particular thing and this drug that you're on. So you may want to be careful with that. And there's even some evidence to say that like certain dosages of mushrooms, while most of them do stimulate your immune system, some of them may actually like repress certain aspects of your immune system. So again, depending on like what you're going through, you might want to talk to a doctor or at least dive into the literature and try to get some more data on what's happening here, and just be aware that not everything is going to be positive for everybody all the time. And there's a figure here in the book that I think is pretty important. I'm not going to talk about it too much, but essentially it shows the difference between a polysaccharide profile of wild mushrooms, cultivated mushrooms, and mycelium. And this is basically some data that I ran because I was really sick of hearing people argue all the time about are fruiting bodies or mycelium better. And I'm like, I'm not biased towards one or the other, but currently most of this debate is happening based on an assay people call like the presence of beta glucans. And beta glucans are complex starches that are in the cell wall of the fungi, protecting the membrane in kind of a scaffolding with manoproteins and other stuff. But beta glucans are really the only kind of like readout for the potency of mushroom supplements. And all it really does is say, hey, there's fungal biomass in here. And sometimes people say I have more or less beta glucans means it's better somehow. And in theory, you're going to get more beta glucans in a like mushroom fruiting product than you will get in mycelia. The thing about mycelia, though, it's a lot easier to grow. You can grow more of it faster. And so certain companies have completely bought into the idea that they're just going to grow mycelia. And I think the real thing there is that it's easier for them from a production standpoint. Instead of having to wait for the mushrooms to fruit, you get more product more consistently. But it does seem weird that often those mycelial products are sold at equivalent prices to fruiting bodies. And there's a particular personality who spends a lot of time like advertising his own brand and telling people that his products are, if not equivalent, actually better than like fruiting body products. And I find that kind of disingenuous because he's not using data to say that. And if you look at the studies he's citing, they've essentially paid for studies that make their products look good, which is always a thing you got to look out with any medicinal mushroom thing, is that a lot of times to get good data, a company pays someone to do the research, and then that sort of slightly favors their product, or they write it up in a way that makes their product look better than it actually is in comparison to like the controls and stuff like that. And what I saw from the data I ran, because I did a polysaccharide analysis of 5 different mushrooms. I looked at lion's mane, I looked at turkey tail, I looked at cordyceps ganoderma, or like reishi mushroom, forgetting if I did one other one. Either way, I did it across like 4 different brands, and I knew 2 of those brands were based on fruiting bodies. One of those brands was using both fruiting bodies and mycelia, and then the last brand was using just mycelia on like brown rice stuff. And what I saw across these analyses pretty consistently was that wild mushrooms and cultivated mushrooms had very similar polysaccharide profiles. So it didn't necessarily matter that it was a cultivated version versus a wild version. Those extracts and the not extracts, but the profiles of the polysaccharides looked pretty similar. And then mycelia, and I only had the cultivated mycelia versus wild stuff because I couldn't just go out and dig out random wood, but we had the mycelial products from this particular company. Those looked really different from the fruiting bodies. And the thing that was both fruiting bodies and mycelia looked kind of intermediate between those things. And again, I'm not talking smack about mycelia. I think there's an idea that maybe mycelia might be more bioavailable. The stuff that's in there is in smaller pieces, easier to digest, easier for our bodies to access. However, the composition of polysaccharides is different. So fruiting bodies and mycelia are not giving the same sets of compounds to you when you take them as a supplement. And again, I'm not to say one is better or worse. I think there's probably uses for both of them, but recognize if you're taking mushrooms as medicine, the actual mushroom versus the mycelium is going to be different, and they may have different applications or different effects. And that's all I really wanted to drive home with this experiment that I did for the book. And I've thought about trying to talk to some of these companies that sell supplements and get them to buy into looking at this type of polysaccharide analysis, or even just show them the data and say, hey, I just want to sit you down and show you this data and maybe have a conversation with you guys. And I think there's one company in particular, the one I sort of mentioned, that should retool the way they do some of their advertising, because it is disingenuous to somehow constantly say that their mycelial products are better than fruiting body products. But that's my soapbox for another time. So let's keep reading. So this says, what are some medicines that we have derived from fungi? A broad array of incredibly important drugs and therapies have been derived from fungi. One of the most famous examples is the first antibiotic ever discovered, penicillin, derived from Penicillium mold, the same genus found in blue and on brie cheeses. Another famous example is Paclitaxel, or Taxol, an anti-cancer chemotherapy drug that inhibits tumor growth that was originally isolated from Pacific yew trees and is produced as a secondary metabolite by fungal and bacterial endophytes. Statins are another common fungal-derived drug prescribed to lower cholesterol and combat heart disease. Statins were originally isolated from Aspergillus terreus, a common soil mold, but are also found in many mushrooms, especially oyster mushrooms, pleurotus. There's a bunch in shiitake too. Another extremely important drug is cyclosporin, a potent immunosuppressant that is necessary for successful organ transplants. It was isolated from Tolypocladium inflatum, a cordyceps-like mushroom that parasitizes scarab beetles in its teleomorphic sexual stage. This fungus also produces small proteins called ephropeptins, which are utilized for their antifungal and insecticidal activity. These are just a few examples of ways in which studying mushrooms and fungi have had real positive impacts on human health. The deeper we dive into the chemical toolkits of fungi, the more we can discover novel compounds that drive medicinal or medical innovation and improve our lives. So, in the whole raging debate over medicinal mushrooms, I think it kind of gets lost, the fact that we have actually developed a lot of drugs and therapies, antibiotics in particular, using fungi as our toolkit to look into what they're making, and then understanding some of their activities. Like I talked about the cyclosporin, that's produced by this Tolypocladium thing as a mechanism for it to invade and repress the immune system of what it's infecting. And we're using that same logic, but we're using it in a medical context to help people from rejecting an organ transplant. So you don't like reject your liver or kidney after getting it implanted. And I just think that's really cool, because instead of sort of looking at medicinal mushrooms as this like hand wavy hippie kind of thing, we can actually say, hey, here's all these like really well-defined concrete examples where science has isolated and understood a molecule produced by fungi, and we're now using this as a drug and a therapy. And it's, penicillin is probably the most famous and important antibiotic of all time, and has literally saved millions upon millions of lives, and started the whole sort of antibiotic revolution, which, even if you have mixed feelings about that, is still incredibly impressive and was a massive step forward for modern medicine, potentially sort of enabling all of modern medicine, by the fact that we had something that could fight bacteria. Okay. How are medicines derived from fungi? And this is, again, very different from sort of medicinal mushrooms and supplements. But first, extracts of wild fungal isolates are screened for biological activity until a compound is discovered. And this is the kind of like bioprospecting stuff they do. Sometimes, someone will go to the jungle, pick a bunch of leaves, and then they isolate endophytes from those leaves, and then they run them through all sorts of tests to see if they produce interesting chemical molecules, Okay. To turn a bioactive compound in a pharmaceutical drug, it is necessary to produce it in a lab under controlled settings. To do this, the cluster of genes responsible for making a compound are isolated and added to an expression system, like Pichia yeast, which is grown and processed under specific conditions to obtain the pure compound. So basically, you can like take a gene cluster out of some fungi that's making it, but is maybe hard to culture, pop it into a yeast, which is really easy to grow and induce in particular ways. And then from that, you create a bioprocess, which you're able to, like, in the end, isolate a molecule or drug, which then goes on to be the therapeutic agent. Alternatively, once bioactive molecules have been fully characterized, they can be produced from precursor compounds via organic chemical synthesis. And that's another thing too. So once you discover it in fungi, if it's difficult to make a lot of it biologically, you can just find some sort of chemical precursor and put it through a couple chemical steps to like tack on a few extra moieties or turn it into the drug of choice. And sometimes they even take the original like framework that fungi did, and then they add a few extra things to it to make it a proprietary drug. And that's how a lot of drug companies work, is they do natural product mining, but then they like slightly chemical modify something, which they can then patent, because you can't just patent natural processes. For fungal derived compounds to get approved as a drug, they must go through a multi-stage clinical trial and receive FDA approval. This process requires significant time and investment, but ensures the safety and efficacy of a therapeutic agent, a fundamentally different process from the way mushroom supplements are produced and marketed. So I'm throwing shade at the mushroom supplement industry because there's no rules, there's no guidelines, there's nothing that says their product has to actually do what it says it does. But if you're going to make a drug and advertise it, it better do what you say it does, and it better have been screened for side effects, and people better have an idea of like what can go wrong. And that's not necessarily there for stuff like supplements. How does eating mushrooms stimulate our immune system? Mushrooms can help support a healthy immune response, acting primarily to stimulate the various components of our immune system. The beta-glucans function directly as immunomodulators. As they get digested in our intestines, pieces of polysaccharides are absorbed through the lining of our intestines, the lumen, and co-opted by our white blood cells. These pieces are also incorporated into antibodies and help to train our immune system to recognize and fight off foreign invaders. So the white blood cells are also catching pieces of those fungal sugars and starting to recognize them, which helps them. If there is a fungal invader, they're on the scene. They know how to grab it because they've seen it before. And that's part of the immunomodulatory, stimulatory effect that eating mushrooms can have. Additionally, mushroom polysaccharides have been demonstrated to stimulate the production of cytokines, small proteins that regulate our immune response and hormone function. The polysaccharide lentinan, derived from shiitakes, has been studied for its ability to stimulate white blood cell growth and function, helping our immune system fight cancers, resist viruses, and recognize microbial invaders like malaria. Besides polysaccharides, mushrooms contain a bevy of other bioactive compounds that can have stimulatory effects on our various types of white blood cells and our immune system as a whole. So there's certain types of sugars that will stimulate certain types of white blood cells, like leukocytes, or there's a whole bunch of different white blood cells. I couldn't get too specific in it because it's a really deep subject, and I'm doing my best to kind of summarize things at like a surface level here. But I do cite my sources. So if you want to read these papers and dive deeper yourself, you can go to the reference section of the book, which is a QR code, and you can go to my website and then look up all of these papers that I'm referencing and read through them if you're interested. How can you tell if a mushroom is toxic? Poisonous animals often advertise their dangerousness to potential predators through flashy colors. There's a name for that. I forget what it's like, the aphiomorphism or something like that. However, for mushrooms, color is not a dependable indicator of either toxicity or edibility. Overall, only roughly 3% of known mushroom species are considered toxic. Many genera of mushrooms contain both edible and toxic species, which can be deeply intimidating when you're first getting familiar with them. Some edible species also have toxic lookalikes, making proper identification challenging. The best way to differentiate dangerous mushrooms from edible ones is to look at many examples of both. Focus on learning the core traits, like spore color, morphology, habitat, etc., that will help you confidently identify different genre and species. And I reference you to a page where I talk about toxic mushrooms later in the book. The only foolproof way to avoid toxic mushrooms is to learn each species individually and collect only the mushrooms you can identify with absolute certainty. If you do want to collect unknown mushrooms for scientific purposes, keep them in a separate labeled container from your edible mushrooms so there's no chance of cross-contamination or confusion. And this is also when I was in the foraging chapter, I was saying, wax bags are not useful for collecting lots of edible mushrooms. But if you want to pick up a mushroom to bring home and study and better understand it or compare with books, throw that in a little wax bag, and that'll stop it from touching the other stuff. Because some of the toxins in mushrooms, while they don't cross through your skin, can get onto another mushroom because they're water soluble and then get into that mushroom. And you wouldn't want to accidentally eat a mushroom that has been like lying right beside a death cap. That would be bad news. So again, if you want to study stuff, good idea to put in a bag, keep it separate. Should I be afraid of poisonous mushrooms when foraging? The scariest part about poisonous mushrooms is the unknown. Learning about the different kinds of toxins in mushrooms is essential to helping you understand the risks associated with foraging or even cultivating your own mushrooms. The toxicity of mushrooms is not a binary yes-no. Some poisonous mushrooms are deadly from one exposure, while others require a threshold amount to be consumed or repeated exposures to cause detrimental effects. The majority of the toxins in mushrooms are secondary metabolites that are produced in a species-specific manner. If you do want to forage, it's wise to understand the toxicity and relative degree of danger posed by these fungal poisons. Some toxins in mushrooms can be mitigated by special preparation, while others are heat stable and remain deadly even after they are cooked. Some of the toxic compounds in mushrooms have been adopted as medicines, but only as purified drugs that are prescribed by a doctor. Do not attempt to self-medicate with mushroom toxins. And that's a really important point because somewhere in here, I think I talk about the fact that certain amatoxins have been used as chemotherapeutic agents, and they're able to very directly target cancer cells with amatoxins and kill those cells. But if you have cancer, you don't want to go and just eat death caps. That's a very bad idea. That is not how it works. So I'm cautioning you to not just self-medicate with toxic mushrooms. Okay. Can you be poisoned by touching a mushroom? The majority of toxins found in mushrooms do not diffuse across skin. This means mushrooms are generally safe to touch, including toxic species. That being said, some mushrooms can cause rashes for sensitive individuals after touching or handling. For example, the slime on jacks or swillus mushrooms, slippery jacks, can have kind of a goo on top, and I've heard of some people getting a rash when they touch it. Or the mature insides of an earth ball, scleroderma. They're kind of black and purple, and they can also cause a little bit of a rash if you touch it too much, and only, again, for certain people. In Asia and Australia, and basically the whole Pacific Rim, there is one particularly evil-looking mushroom called the fire coral, otherwise known as Trichoderma cornudemae. This species does contain skin-permeable satrotoxins. It's the same stuff that's in newts, and can reportedly cause severe skin irritation when handled extensively. And people have died from eating that mushroom, but I don't think anyone's died from touching it, and the worst that can happen really from touching it is getting kind of a severe rash. Regardless of what you've touched, you should wash your hands thoroughly after mushroom hunting. That's always good advice. If you're out in the woods, wash your hands when you get home, or even before when you get back to the car. If you have particularly sensitive skin, you can always wear a pair of gloves while you're out foraging. And I do that sometimes. Even I don't have sensitive skin, but when I'm in the mountains and I'm sticking my hand into the dirt a lot to pick mushrooms up out of the dirt, I wear gloves sometimes because I get all sorts of dirt, and the soil is pretty basic, so my hands get really dried out and crack. And so if I'm doing several days of mushroom hunting, and especially camping, once you get dirty hands while you're camping, they'll never get clean. So sometimes I do wear gloves when I'm mushroom foraging just to keep my hands clean and have less of a chance of irritation or rash or just dry skin. But I'm not worried about touching the mushrooms. That's the big thing. How long does it take after eating a toxic mushroom to get sick? The most common type of mushroom poisoning is acute, with symptoms appearing soon after consumption, usually within 20 minutes to about 4 hours. However, the most dangerous type of poisoning can have delayed onset, with symptoms manifesting within 6 to 24 hours or even several days to weeks later. This can make it difficult to determine what the cause of poisoning was. Some toxins may not have any immediate effect, but can build up over time, causing chronic symptoms or health problems. As with many things in life, this is in quotes, the dose makes the poison, meaning small amounts can be tolerated, but larger amounts may have serious health consequences. Many of the toxins in mushrooms are not immediately poisonous, but are processed through a digestion and metabolism into a form that is problematic. This explains the delayed onset and chronic nature of some symptoms. So the mushroom itself may have a compound that's sort of like a proto-drug, and it's not immediately toxic, but if you eat it and consume it and it gets into your liver, then enzymes start like breaking it apart, and suddenly one of those pieces that's been broken apart is toxic and gums up the works in your insides, and then you get sick, but it's like, many hours to days after you've eaten the thing, which makes it much harder to say like, what's wrong with me? Because you might not even remember that you ate the thing. And that's the real fear with mushroom poisoning, is one of these like delayed onset things, because those are often associated with some degree of like organ failure, which is bad. Most mushroom poisoning is like, oh no, I got diarrhea, and you'll get over that in like 12 to 24 hours. But you won't get over your kidney dying. that's bad. Then you need to get yourself an organ transplant and take cyclosporin. So, okay, what are the effects and symptoms of different kinds of mushroom poisoning? Gastrointestinal, or GI, and I'm going to say the word GI a lot, but the most common symptom of mushroom poisoning is gastrointestinal irritation, usually with acute symptoms including nausea, cramps, vomiting, and diarrhea. GI symptoms can be particularly distressing, but will generally resolve within 24 to 48 hours, rarely requiring medical intervention. Polysaccharides are often the culprit of GI distress, usually due to undercooking, overindulgence, consumption with alcohol, or just personal sensitivity. So again, if you get the shits from a mushroom, it's probably not that big a deal. You can go see a doctor, but likely they'll tell you just let it pass, kind of thing. Okay, neurological and physiological. Other than GI symptoms, mushroom poisoning can present other types of physiological symptoms, including but not limited to shortness of breath, sweating, salivation, irregular heartbeat, chest pains, skin rashes, tingling, flushing, and anxiety/nervousness. Excuse me. Sorry. Some of these symptoms can be caused by co-consumption of other toxins, especially alcohol. So I cautioned at the end of the edible chapter to be careful about consuming a lot of wild mushrooms and alcohol at the same time, because sometimes your body can't process the stuff that's in the mushroom as well as process the alcohol. That's especially true of stuff like morels and verpa, or two mushrooms in particular. I've heard people having issues with alcohol, even chicken of the woods, maybe. Physiological symptoms still presenting 24 hours after consumption are a sign of more serious poisonings. Seek medical advice and treatment if you experience persistent symptoms. A less common but no less scary type of mushroom poisoning is related to our central nervous system, or the CNS. Toxins that affect the CNS tend to present within 6 to 12 hours and cause neurotoxic symptoms, including but not limited to headache, disorientation, delirium, seizures, and rarely death. These hydrazine toxins usually leave the body quickly, with symptoms resolving on their own. However, you may still want to seek medical attention, as even one acute exposure to hydrazines can have long-term health effects. They can be carcinogenic. So if you do have a hydrazine poisoning event, it's probably not a bad idea to go check with your doctor, even if it resolves and you end up feeling fine on your own. And hydrazines are stuff primarily present in the gyromitra, toxic, sort of what they call false morels, or lorchels is what I call them in the book. There's also some hydrazines in some agaric mushrooms, which are like relatives of the portobello, but they're not in cultivated portobellos. and that's a piece of misinformation that's been propagated via like the Joe Rogan podcast. Cultivated portobellos and creminis and white button mushrooms are all safe to eat raw. I don't like eating them raw. I don't think they taste super good, but they're safe because they've been cultivated and selected to be safe for like many, many years. If you look at wild agarics that have not been cultivated or grown, selected for many years, those can contain like a appreciable amount of hydrazine. So it's not a hazard if you cook it because hydrazines blow off. They volatilize really easily. But if you eat them raw, you could potentially poison yourself. And hydrazines are weird too because they have to reach a certain threshold. So you can eat a fair amount and not feel sick. But if you have like one too many mushrooms, suddenly you're like all disoriented and really messed up. And it's kind of a scary toxin. And it's one that can kill you. So that's part of why I'm talking about it. Okay. Delayed and chronic. Delayed or chronic symptoms can be signs of more serious, potentially life-threatening poisonings that require medical intervention to prevent death. The delayed onset of symptoms is caused by toxins that are metabolized and cause severe organ damage, particularly to the liver and kidneys. For example, amatoxins and orlanine, which is in what am I trying to say? Cortinarius, making it the most severe and scary type of mushroom poisoning. These delayed onset ones. Treatment options for these types of toxins are limited. The consequence of consuming these toxins is often permanent organ damage or death. Be extremely careful and aware of the risks if you plan on consuming any kind of amanita or webcap, a Cortinarius, which can contain these toxins. Okay. And one thing that really annoyed me is in a lot of books, they include psychedelic mushrooms as toxins. And so I have them in this section just because that's sort of the convention, but I don't view psilocybin as a toxin. However, were you to like consume it without prior knowledge to what it was or what its effects were, it would feel very much like you had been poisoned. And I think that's part of why it's always been included in like a toxic mushroom section in the book. But here I gave them their own like mini section. So I said hallucinogens and entheogens. Although they are often thought of as drugs rather than toxins, the hallucinogenic and entheogenic compounds in mushrooms can cause symptoms of poisonings if consumed without prior knowledge or intention. Consumption of compounds like psilocybin and muscimol affects the CNS and can present a number of symptoms, including but not limited to hallucinations, euphoria, dizziness, time distortion, drowsiness, coma, GI upset, and mood instability. These compounds are not life-threatening and generally do not require medical intervention. So if you eat too much, you don't need to go to a hospital. In fact, that would probably make it a very bad trip. So just chill out, take a deep breath. I talk about that later in the book. And I have a little sidebar here where I talk about what should you do if you suspect an incident of mushroom poisoning. So I'm just going to read you the advice here. Hopefully, you'll never have to deal with a mushroom poisoning event. But if it happens to you or someone you know, here are the steps you should take to ensure the safety of anyone who has consumed a potentially poisonous mushroom. Call poison control. I give the number for that. You can just Google poison control. Call them. Ask them what to do. Take a picture of the mushroom and save a specimen, even if it's already been cooked. This can be used by experts to identify what species and specific toxins may have been consumed. Consult with online mushroom poisoning forums or groups on social media. There's a really good one on Facebook called Poison Help and Emergency Identification for Mushrooms and Plants. And there's a lot of experts who are on there, and you'll usually get a response within like 5 minutes or less. Drink a lot of water and stay hydrated to dilute the effects of the toxins. That's a thing to do if you think you've been poisoned. With a doctor's supervision, consider taking activated charcoal. If the symptoms are severe enough, call 911. In cases of suspected amatoxin or orlanine ingestion, seek immediate treatment, including intravenous fluids and dialysis. And that's because these things, once they get into your system, can be toxic for a very long time, and they're hard to get rid of. So if you ate one, usually within about 24 hours, they can help flush a lot of it out and vastly improve your chance for survival or not having organ damage. But if you wait too long, then there's not that much they can do for you. Okay, and if you have it, use an amatox test kit to detect the presence of amatoxins. And I have a little QR code. Basically, it's like an antibody test or like a COVID test for this amatoxin thing. And you can buy this as a commercial product. So if you're freaking out because your dog or your child ate a mushroom in the yard, you can keep a couple of these little test strips around, and you can immediately see if, okay, this doesn't contain amatoxins. I'll keep an eye on my dog, I'll keep an eye on my kid, but at least this isn't like a deadly toxic mushroom. Whereas if it does contain amatoxins, it's like you got to get to the hospital like right now. Call the poison control, call specialists, get on the horn, and go do something about it as fast as you can. So having that little test around can really help your anxiety if you're worried about, the random mushroom that, your dog ate while you're on a walk. Probably wasn't a big deal, but just in case, that test kit can help you be sure. Okay. What are the risks of mycotoxins in food? And mycotoxins are really like deep, complex subject. I sort of just scratched the surface here, but these are important things that we don't talk about that often because we think about toxins in mushrooms, but we don't understand sort of the ever-present danger that mycotoxins are posing to us, partially because our food system is pretty safe and pretty good, and we don't see them or have to deal with them. But I do think that there are some like health problems out there that people are unaware of that may be caused by the unknown consumption of mycotoxins. Okay. Common soil microbes like Aspergillus and Fusarium species can produce a wide range of extremely dangerous mycotoxins depending on the particular species and specific growth conditions. These kinds of toxins are usually consumed unintentionally as a byproduct of microbial contamination during food storage and processing. The most common food-associated mycotoxins or foods associated with mycotoxins are cereal, grains, corn, nuts, seeds, fruits, coffee, and spices. The effects of these toxins are often less acute than those found in mushrooms. They're sort of like low-lying, like background effects. But instead of chronically severe and particularly notable, but instead they are chronically severe, which means they can cause issues over time, and particularly notable for their carcinogenic, cancer-causing effects. So a lot of mycotoxins don't necessarily make you sick immediately unless you consume a very large amount, and you don't normally do that unless you're eating like moldy bread. But if you have like a tiny bit of mold on your bread that you don't notice or you scraped off, you might still be consuming small amounts of mycotoxins, which even small amounts can be chronically severe if you do it many, many, days, years in a row. And even those small amounts can be carcinogenic in that they're starting to cause DNA damage that may eventually lead to cancer. So that's part of why it's really important to understand mycotoxins and minimize your risk and exposure as much as possible. And a lot of that happens through our food system, but it's helpful to understand better where the risks lie. So 3 of the most common classes of mycotoxins are aflatoxins, ochratoxins, and fumonisms. Fumonisms? Have been shown to cause oxidative damage to DNA, leading to increased cancer rates in humans and animals alike. These mycotoxins are notorious for causing serious epidemics of foodborne illness for humans and animals throughout history. One example is the outbreak of ergotism caused by Claviceps purpurea on rye grains, and this is the historical cause of St. Anthony's fire and has led to countless deaths throughout recorded history. It's the same kind of thing that caused like the Salem witch trials and stuff too. It was the outbreak of ergotism and people consuming the ergot, which is the claviceps stuff, and it grows on grain and it gets into the bread, and then a bunch of people might have been consuming sort of LSD in their bread without knowing it. And women in particular are, because of like reduced circulation, there's a number of physiological effects that makes them more prone to experiencing the side effects of like ergotism and this poisoning. And so that's part of the idea of like where the Salem witch trials came from. Was just a food safety outbreak basically, from contaminated grain. In the 60s, mysterious deaths of more than 100,000 turkeys due to turkey X disease led to the discovery of aflatoxins in their feed, which is produced by Aspergillus flavus. And during the First and Second World Wars, outbreaks of alimentary toxic aleukia among troops led to the discovery of grains infected with fusarium that produced the T2 trichothecene mycotoxins. And so I have a big sort of figure here in the book of some common sort of molds and things that are just ubiquitous all over the place, the foods they infect, and then some mock-ups of the actual mycotoxins that are produced, and then it can affect GI upset, carcinogenic properties, can affect your central nervous system, can cause organ damage, and even suppress your immune system. So I think there's a lot of potential bad stuff that can happen from mycotoxins. And for the most part, we're sort of ignorant to it because we don't pay attention to it, and we just assume our food is safe. And for the most part, it is. And here, I'll read you my answer. Should I be worried about mycotoxins in my food? The presence of mycotoxins in our food represents a major threat to human health, but thankfully, advanced food processing technologies help to separate contamination, greatly reducing the risks. In the USA, the FDA sets allowable limits for and mandates testing for certain mycotoxins in food products. These regulations reduce the danger posed by these mycotoxins for processed packaged food, but there can still be risks associated with mycotoxins depending on your diet exposure and personal sensitivities. A great example of this is that pretty much all peanut butter has some base level of aflatoxin in it. So if you eat peanut butter all the time, and it's like one of the primary foods you consume, you're eating probably more aflatoxins than you should be. I mean, I don't think it's that big a deal if you have peanut butter every once in a while, but if you're doing it like consistently, if you eat 4 peanut butter sandwiches every day for lunch, you could potentially have health problems that are being caused by the exposure, chronic exposure to aflatoxins. And it may even have something to do with like food allergies too. Who knows? Unfortunately, many developing nations lack the technology and testing necessary to ensure their food stocks are not contaminated with mycotoxins. Global commerce and movement of goods between countries means that food contaminated with mycotoxins can still pose serious health risks for human and livestock worldwide. So the way that grain gets moved around the world is we might test something here, but if a batch of grain or something has skipped between a couple different countries, we get it and someone is lax on the testing and just like throws it right into a product, there could be mycotoxins in your food that you don't know about. And that's kind of what scares me. It could even be affecting like livestock and animals. And that's a hard one too, because you want to take care of animals, but if you end up feeding them food that's contaminated with mycotoxins, you might be hurting them with the food. Okay, so this chapter ends with sort of a big table of mushroom toxins. And I really wanted to have like a cute little infographic here in the book that was just like a mushroom and a toxin, and I would just kind of like cover different categories of stuff. But it turns out that the toxins in mushrooms are really complicated. And I tried to do a whole podcast episode about this at one point and got like very much into the weeds of trying to describe this stuff. And it was hard, but this table is sort of the best summary I have seen anywhere of all the different types of toxins. And I'm going to very quickly try to like discuss at least a few of them. And again, these are graded by the spectrum of threat that they pose. And this is how I've organized the toxic mushrooms in the book, is basically calling them deadly, dangerous, or sort of beware/caution. And this is, one approach. I'm not going to say it's the best, but I think it's helpful for you guys to recognize that some toxins are like deadly, some of them are dangerous, and then some other things you should watch out for, but probably aren't going to kill you or really, cause anything other than GI upset. Okay. So I start out with an example of proteins or peptides, which are deadly. And these are amatoxins. You find them in like the death cap mushrooms and destroying angels. They're in section Phalloideae for those amatoxins. They're also produced in some Galerina, phenolbells, as well as in some Lepiota, which are dapperlings. And so the onset speed is about 6 to 24 hours until you get sick. So it can take a little while. And the duration is about 4 to 10 days. If you don't seek treatment and you've eaten enough, you'll be dead at the end of those 10 days. And it's a very unpleasant death. It's awful. All of your organs start shutting down because the amatoxins bind irreversibly to your RNA polymerase II, which is what turns DNA into RNA in your liver. And so your liver stops making new protein and it just starts melting basically. And then the rest of your body follows. So there's not a lot they can do for amatoxins. There are some emerging things like milk of thistle and this like indocyanine green. There's things they can do, but all of them have to be administered usually within the first like 24 hours. And again, that's one too, where drinking lots of water and trying to flush your system as much as possible can greatly improve the health outcome. Okay, so a protein or peptide that is dangerous. An example of this is bolicitine, which is in Satan's boletes or rubroboletus. And this inhibits the sort of protein synthesis as well as like potentially causes blood clumps. It's not usually deadly toxic, but it has been known to cause, a fatality before. Its onset speed is usually about 2 to 4 hours, and you can get like hypertension, dizziness, bloody diarrhea, organ damage. But you can treat it by doing like blood thinners, and it's usually going to last about 6 to 24 hours. So it's not as dangerous as an amatoxin. That being said, I don't eat Satan's bolete, and I've talked to some people who swear by like boiling it 3 times and then eating it. And I'm like, bolicitine is a heat stable compound. I'm not comfortable consuming something that is still toxic. Whether or not you can dilute it out of the mushroom is questionable to me. So I would not recommend eating Satan's boletes. And I use Satan's bolete as a common name for rugoboletus, which is a thing that some other people take issue with, but this is how I do things. So an example of a protein or peptide that you want to be aware of would be called something osteolysin, which is in oyster mushrooms or pleurotus. And the mechanism of action is that it kills cells. It's cytotoxic by poking holes in the cell membrane. So it's something that kills red blood cells, and that's not good for you. The onset speed is about 2 to 4 hours, and the duration is about 24 hours. And this happens from eating raw oyster mushrooms, which I don't recommend doing. If you eat a lot of them, you can get sick. Fever-like symptoms, GI upset, rash, weakness, headache. You can treat it by doing fluid IV and like supportive care. This is just one of those things you kind of get over because it's, beware, caution rather than like deadly or even dangerous. Let's see. We're going to talk about slow onset kidney damage. This is a scary one. So this is the thing that orlanine does. It's in deadly webcaps or Leprosopy species. So it inhibits DNA RNA synthesis and can cause DNA damage. So it can also be carcinogenic. Its onset speed is very slow, like 2 to 14 days. And the duration can be 21 to 30 days. And so initial is like flu-like symptoms, and the delayed things would be kidney failure, lack of urination, and it can be fatal if untreated. And what you can do is you can do some fluid dialysis, blood dialysis. There's some types of steroids you can take and just sort of provide supportive care. And that's a very rare form of poisoning because people don't really eat Cortinarius, and the deadly webcaps are not super common. So you don't hear about very many poisoning cases happening with this particular thing, but is one of the scariest types of mushroom toxins that exists. I talked a little bit about hydrazines, but those are neurotoxins. They can be deadly. It's monomethylhydrazine is the form that's usually in mushrooms that is dangerous to us. And they're in stuff like lorchels or gyrometra, and even some agarics. They attacks the central nervous system, stops production of essential neurotransmitter GABA. So the onset speed is about 4 to 16 hours when eaten, or 2 to 8 hours if inhaled, because it can be volatile as well. And duration is about 2 to 6 days. Symptoms are severe GI upset, fever, dizziness, lack of coordination, vertigo, tremor, headaches. In some rare cases, the symptoms are latent, and you can get serious complications like liver, kidney damage, seizures, not getting enough oxygen, respiratory arrest, and even death. And so there's not a ton they can do to treat that, but you can do activated carbon, get some vitamin B6, and have supportive care. So it's probably best to avoid eating gyrometra. And there are some recently reclassified gyrometra that are now like Mount Blancomyces or neogyrometra. There's like other names for them, which helps differentiate them from the toxic gyrometra. And those other ones are typically good to eat and don't contain any of the hydrazines, but actual gyrometra do. So be very careful with those. Another one I'll talk about because it's relevant would be neurotoxin that is dangerous. And this example is muscarine. And so muscarine is in mushrooms like fool's funnel or Clitosity ribulosa. It is in Amanita pantherina as well. It's not actually in Amanita muscaria at any appreciable amount, but it is fairly like big component of Amanita pantherina. And there's people out there who talk about eating that mushroom as if it were like a psychedelic mushroom, while disregarding the fact that if you eat it, you're going to get muscarine poisoning and you'll just be like drooling and feel like ass. So I don't understand that, but whatever. Okay, so muscarine mimics acetylcholine, affects the parasympathetic nervous system. Its onset speed is fast, about 15 to 30 minutes, and duration is about 2 to 6 hours. Causes blurred vision, excessive salivation and sweating, tears, trouble breathing, cramps, diarrhea, low blood pressure, and in rare cases can lead to heart attacks. And you can treat this with something like atropine or scopolamine, but I don't recommend trying Amanita pantherina. That's not a good idea unless you want to poison yourself. Okay, speaking of Amanita muscaria, Another neurotoxin that you should beware / be cautious about, but is not necessarily deadly toxic, is ibotenic acid. And this is what's in fly agaric Amanita muscaria as the prodrug to muscimol. So ibotenic acid is an agonist of NMDA glutamate receptors. Its onset speed is about 20 to 30 minutes. It's pretty fast. Duration is about 2 to 24 hours. It can induce seizures and cause lesions in specific brain regions, inhibits learning and memory. And so this is why I get really nervous every time I see people talk about Amanita muscaria on the internet, which there are safe ways to process it. You can double boil it and extract the toxins out and then just eat it as a normal mushroom, or you can dry it out and use UV exposure or even low pH to help you finish the chemical conversion, the decarboxylation of ibotenic acid into muscimol. And muscimol is a relatively safe, but it's still a GABA agonist and acts a little bit like alcohol. So if you have too much of it, you get the spins and you don't feel good. But ibotenic acid itself can actually cause potential brain damage. And so there's a lot of people out there, every time there's an Amanita muscaria video, someone's like, eat it raw. And I'm like, no, don't do that. That's bad. And I don't know, it's frustrating to sort of see people like giving really dangerous potential medical advice on the internet as if it were like a joke. And as I love joking about mushrooms, but I don't find jokes about toxic mushrooms funny because people are like perpetuating dangerous misinformation, and that makes me really nervous. So those are one of the few things I don't joke about is the toxicity in mushrooms. I'm sort of dead serious about it because you have to be, otherwise people get the wrong idea and could do something that would be dangerous. Let's see. I'll talk about just a couple more. I don't want to go through all of these, but let's talk about gastrointestinal irritation and allergic reactions. This is dangerous. So an example of this would be toxins in jack-o'-lantern mushrooms, Omphalotus, called illidins. So these can cause oxidative damage and potentially lead to DNA damage as being a carcinogen. Onset speed like 10 to 30 minutes, duration of 12 to 24 hours, cause severe GI upset, nausea, vomiting, diarrhea, cramps, headache, weakness. And this is what's in jack-o'-lantern mushrooms, which people often mistakenly consume when they think they have chanterelles. And from what I've looked at with these illidins, and there's also these omphalotins kind of things, these toxins could potentially, like, they don't make you sick, they do make you sick immediately, they may also cause like long-term DNA damage. So it's not something you'd ever want to get repeated exposure to. And it's also something you don't want any exposure to, period. So be really careful when you're hunting chanterelles to understand the difference between like a jack-o'-lantern versus a chanterelle. They're pretty easy to tell apart, but it does help to look at lots of photos and get extra opinions. And I've seen people on the internet sort of joking like, oh no, I spent all night in the toilet because I ate jack-o'-lanterns. And you're like, well, you might have also like given your kids cancer by doing that. So be careful. That's why I rate it as a dangerous toxin. Here's one, alcohol interaction. And this is dangerous. Example of this toxin is coprine. And this is present in ink cap mushrooms, things that are in Coprinopsis. So its mechanism of action is that it inhibits alcohol dehydrogenase, the thing that detoxifies alcohol, and causes a dangerous buildup of acetaldehyde in your system, which causes all sorts of like flushing and other stuff. So the onset speed is fast to medium, 30 minutes to 4 hours. And this is basically like if you eat ink cap mushrooms, you now have coprine in your system. And for the next like at least 24 to 72 hours, you really should not have any alcohol because this thing can hang around and inhibit your body's ability to break down the alcohol. And if you consume alcohol while you have coprine in your system, you can get flushing, nausea, dizziness, vomiting, diarrhea. You feel real bad. So just don't drink any alcohol for about 72 hours after you eat an ink cap. And that's not true for the shaggy mane ink cap because that one does not have coprine, but most other ink caps do. So this is part of why I say it is a dangerous interaction to be aware of. The last one in here, I'm going to talk about what I think is pretty cool. This is something you don't see in most books, but I learned it through researching stuff, and I think it's really interesting. Some mushrooms can have a blood thinning effect. And so this is a polysaccharide that is in auricularia or wood ear mushrooms. And so its mechanism of action is that it inhibits blood clotting factors, specifically thrombin, and the effects are cumulative with repeated consumption. So the onset here is slow, from like hours to days, because you often have to eat wood ear mushrooms many days in a row, and you have lots of them every day. But it can then last for up to like 3 to 24 days in terms of this anti-clotting effect. And you can have bruising, excessive bleeding, and unusually heavy menstruation. And there's sort of a working theory. I don't know if you guys ever heard of Chinese restaurant syndrome. It was something that, like, was in the news in like the 80s. And the theory was that people were eating so much Chinese food and there was so much MSG in their Chinese food that they were getting sick and having all these issues. And as far as everything I've read and understood, food safety wise, etc., MSG is just the salt of glutamate. It's in our food naturally. It's typically not a big deal to add it into your food. I think people shouldn't be eating lots of processed foods with tons of MSG in it. But for the most part, you're not, like, Chinese restaurants are using so much MSG that it's problematic. And this whole thing was really just kind of based in racism more than anything else. And what may have actually been the causative agent of people experiencing what they call Chinese restaurant syndrome may have just been from eating so much wood ear, because there tends to be a lot of wood ear in Chinese takeout. And I think if you eat it every single day and that's all you eat, that could build up and become problematic. And so Chinese restaurants got blamed for MSG when it was actually the polysaccharide in auricularia and wood ears that may have been the causative agent there. So I think that's kind of interesting. So I will end on that. Thank you guys for listening to me read to you Chapter 7 of Passport to Kingdom Fungi. Again, if you want to support me or help me out, it'd be really great if you could buy the book, digital or physical copy. You can buy it through the Napa Book Mine if you want a signed copy. I also have the companion field guide and journal, which is a great one if you want to sort of bring this out into the woods with you. That book also has a few extra mushroom profiles in there. If you want to support me, you can subscribe to my Patreon, watch my stuff on YouTube, go to my website, buy Original Fungi, Fascinated by Fungi merch, or just, like my content, interact with my stuff. It would mean a lot to me if you guys could leave a rating or a comment on this episode, and again, share it with your family and friends. So until next time, stay fascinated by fungi, friends. Bye.